All right, so enough about energy and the energy molecule and all that. Let's get into the respiration side of things. There are two types. All right, the first type is aerobic respiration. Aerobic, aero meaning air, needs oxygen to function. All right, and it's going to make about 36 ATPs for every one glucose molecule. We're going to try to follow the 36 idea as best we can, but just so you're aware, in case in your reading you come across something a little bit different, that's okay. Right? And absolutely correct. The anaerobic without oxygen. Right? So unfortunately, this only makes 2 ATPs for every one glucose molecule. Right? So a lot less efficient, and we'll look at this later, but we're going to focus on the aerobic side of things starting out. All right, so you should be able to respond to a few questions here, like where does cell respiration take place? Most of it will take place in the mitochondria. Yep. It also takes place, part of it will take place in cytoplasm. Yeah. Yep. So the overall cell respiration process, I mean, if you say it takes place in the cell, that's that's correct in a general sense. More specifically, the first stage will take place in the cytoplasm. The other 2 will take place in mitochondria. Yep. And let's see, what else? What's the main difference between aerobic and anaerobic? In aerobic respiration, here's the overall equation for aerobic respiration. Right? You can include this at the bottom of the current page or put it at the top of the next one. I don't care. I kind of left the space open for you to do whatever you need to do with it. Right? But what are the reactants? What do we start with, and where do they come from? Right? We're going to get glucose from the food we eat. Right? So we could be eating plants. We could be eating animals that have eaten plants. Right? The oxygen that we're going to get from breathing, right? From the atmosphere, because the plants have exhausted or vented it out into the atmosphere for us to, as a byproduct that we can take advantage of. Yeah. So this is coming from the stuff we eat. This is coming from stuff we breathe. Right? Absolutely correct. Okay, so here we go. Whew. An overview of cell respiration. There are 3 main stages that we're going to look at in overview in our time today. Okay, stage 1 is glycolysis. And what we're going to do is we're going to take some glucose and we're going to convert it into a molecule known as pyruvate, also known as pyruvic acid or pyruvic acid. From there, we're going to transfer that into the mitochondria. So this takes place in the cytoplasm, and we get a few ATPs out of that. Not much, but we get a few. Then we're going to move these guys, this pyruvate, into the mitochondria. There is a, step 1.5 that happens between steps 1 and 2. We're not going to talk about it in detail with an overview, but we'll see it when we look at more detail later on. But long story short, we get these molecules into the mitochondria and they enter into citric acid cycle, also known as the Krebs cycle. This cycle is going to take place in the matrix of the mitochondria. So you'll see Neo here, right? Great trilogy though. Check it out. Great trilogy. I personally like the second one the best. So there you go. Because that's when Neo can fly around and stuff. It's cool. Now they can't in the third. But the third is, never mind, forget it. You have to watch it. It's great. Great. Watch it on Blu-ray. Don't watch it on standard def. All right. So in the citric acid cycle, we're going to create a few more ATP. We're going to create some carbon dioxide, and we're going to load a couple high-energy electrons onto a couple molecules for transport. Okay? So those will then eventually make their way to stage 3, which is known as the electron transport chain, or more formally as oxidative phosphorylation. Let's break that name down for a second. Right? It sounds really fancy and difficult, but it's not. Oxidative. To oxidize something is to lose its electrons. Okay? So we have molecules, and as we look at this figure, NADH and FADH2, right? These are new molecules that we have not seen before, but they function just like NADPH. Anyway, they're electron carriers. They're going to give up their electrons, give them up to the electron transport chain. So there's your oxidative. And then phosphorylation, we just learned phosphorylation is to attach a phosphate to something. Well, we're making ATP here. So we're attaching phosphate to ADP. So there's your phosphorylation. It's a fancy name for we're making ATP. Right? We're going to make a crap ton of ATP out of this. Right? Like 32 or 34 of them in the third phase. Right? So in general, what's happening? Stage 1 and stage 2 are kind of like energy priming a bunch of molecules for delivery into phase 3, the electron transport chain, which will generate a crap ton of ATP for us. How will it generate so much ATP? Well, it's kind of like the opposite thing that happens with the light reaction and its electron transport chain. We'll see that in a second. All right, so here we go. 3-stage process. Stage 1, glycolysis. Occurs in the cytoplasm. What do? We take glucose and we split it into 2 pyruvate molecules. This happens in all organisms. Every single living thing that's cellular does this, right? Every cell on this planet does this. Goes through glycolysis, right? No oxygen is needed. Interesting. Here's the catch. It requires 2 ATP to get started. So you pay in 2 ATPs, but you get 4 out the other side. You double your money. All right, you put 2 in, you get 4 out. Overall, you're going to gain just 2 ATPs. One of the new ones is NAD+. Right? When it's energy loaded, it's NADH. Right? Just like NADP+ was NADPH when it was energy loaded. Same idea here. Here's the catch. All right? We load NAD+ into NADH during glycolysis. There's only so many of those to go around. So if they're all energy loaded, glycolysis stops. It can't work. It can't do anything more. Right? It shuts down. So here's where you would see a separation between aerobic and anaerobic respiration. Right? You would be correct in thinking that if we do not have oxygen, there's another step that's going to help free up this NADH. And you're absolutely correct, there is, in the absence of oxygen. But we're going to focus on aerobic respiration, right? So we're going to consider that there's oxygen present, ready to move on to the next step. Okay. Now, before we move on to the second step, we do need to talk a little bit about these new molecules, because I know we made a list with photosynthesis, right? We made like carbon dioxide and water, H2O, we did all that stuff. We also included NADPH and ATP and ADP, all that stuff. Right, so we can add a couple new molecules here. NAD, which is nicotinamide adenine dinucleotide. It's just minus the phosphate, right? Instead of NADP+, it's just NAD+, right? And NADH is when it's energy loaded. What does it do? It plays a key role in moving electrons. It transports electrons. That's what it does. That's its job. It's electron taxi service. Right? It's going to move these electrons to where they need to be, and then actually it's going to move them to electron transport chain at the end of the process. Right? So really, what does it serve as? It serves as a cofactor to help enzymes happen and work, function and work. Right? Now, at the bottom of this, I put that it's kind of like the molecules we saw in photosynthesis, and it is. It behaves much the same way. But I also included in here that FAD+ and FADH2 are very much the same idea. It's just you see them show up in cell respiration, so we should probably mention them in our list as well. Right, so now we should have a good list of all the players we'll see in cell respiration. The only difference between NAD and FAD is the name, like what they're structured like. So this is nicotinamide, this is flavin adenine dinucleotide. Right, the names are not important. What's important is the fact that you remember that these are high energy electron carriers. Right? Right? They're loaded is with H's, unloaded, they're NAD+, FAD+, respectively. If oxygen is present, then this happens. Right? It completes the breakdown of glucose so our original glucose molecule, it gets broken into two pyruvates, and those two pyruvates get moved into the mitochondria, and they get converted into a usable form in this cycle. Right? Overall, what happens? Completing the breakdown of that original glucose molecule, it's going to make two ATPs, and will also supply high energy electrons to NADH and FADH2. Right, so we only get two ATPs out of the cycle, but we do get a significant number of NADHs and FADH2s, which are carrying high energy electrons to the next stage. So I don't know what you want to think of this as. I don't know. The station, the loading station, the loading dock. But it's gonna load up NAD+ and FAD+ with high energy electrons. And along the way, phosphorylate a couple ADPs along the way. Right. So now what we've done is we set ourselves up with a load of molecules containing high energy electrons. So what are we going to do with them? We're going to move them into stage 3, the electron transport chain. We're going to offload these electrons into the ETC. Right, this is going to occur in the mitochondria, but along the inner membrane. It's very similar to what happens along the thylakoid membrane in photosynthesis. What you need to understand is that in photosynthesis, in the thylakoid membrane, along the light reactions ETC, we were pumping H pluses into the thylakoid. Right? We're increasing the concentration of H pluses inside the thylakoid. In cell respiration, we're going to be moving these electrons along the transport chain, along the membrane, to pump H pluses out of the matrix and into the space between the membranes. Right? This energy is used to transport H pluses out of the matrix and into the intermembrane space. Now, simple conceptual question. If we are overloading the concentration of H pluses in the intermembrane space, where do those H pluses want to go naturally? We're pumping them out of the matrix into the intermembrane space, using energy to do that. Where do they want to go naturally? They want to get back to the matrix. Yeah, absolutely true. Right? Well, they are H pluses. They can't go through membranes. So guess what they go through to get back in? Along the inner membrane of the mitochondria are a crap ton of those ATP synthase guys, just waiting for H pluses trying to get back into the matrix. Right? We'll close on this image. Right? Here's what I want you to take away from this last look before we call it the day. Right? This is showing you an image of the a mitochondrial inner membrane. Okay? This is the intermembrane space in the dark orange. This is the matrix in the light peachy color. Right? So these high electron carrying molecules like NADH and FADH2 will deliver their electrons to the chain. And as they move along the chain, they're going to pump H pluses out. Right? And then to get back in the matrix, they have to go through ATP synthase. This is just like what we saw with electron transport and the thylakoid, except it was flipped. Right? Because we were loading them into the thylakoid so they would flow back out into the stroma. Right? This is just the opposite of that. It's rather clever.